welcome to hello and welcome to the second episode of Sci Cam. I'm Michael Conterio, and joining me this week are Emma Pewsey. Emma?
Hi.
And Sonja Dunbar.
Hello.
We're going to start off now with our interview, and my guest is Alex Shires from Imperial. Hello, Alex.
Hiya.
Alex has been doing a PhD at Imperial College London and also at CERN in Switzerland. So could you tell us, what have you been researching?
I've been looking at the effects of virtual particles, which are imaginary particles, in the decays of other particles as they're produced at the LHC.
And what's the big reason behind this? What are you trying to get out of all of these particles?
So as you smash your protons together, you create what we call B mesons. And B mesons have a lot of mass, and as the B mesons decay, they transform that mass into energy, and then back into mass to produce the daughter particles. And in this transition, new particles, whatever they may be, may cause various rather subtle effects, but various effects which we can find out, and we can use the information from the daughters of our parent particle to find out what actually happened and whether there are any strange new particles that happen to interfere at a much lower level.
Okay, and so you're talking about mesons. Can you explain a little bit more about what they are?
So meson is a quark and antiquark. Quarks are the most fundamental particles that we see, along with electrons and this sort of thing.
So you're working with the very fundamental building blocks of the universe and trying to learn more of the universe that way.
So as one set of quarks transform into another set of quarks, strange effects happen, and this is what we look at.
And one of the big strange effects that you're trying to look at the LHC, you're working, not looking for the Higgs boson specifically, right? You're looking at a slightly different experiment.
Yeah. So I'm on LHCb, which is a slightly smaller experiment. You may have heard of CMS or Atlas, which are the large detectors that have, quite probably found the Higgs boson. So I'm working on LHCb, which is a slightly smaller detector, which is very specialised for looking at these B mesons. And as I said before, the exciting thing about these B mesons is that they have a lot of mass, and they can decay in a variety of interesting ways. So these B mesons are our sort of special little corner of particle physics, and the new particles we're looking for sort of show themselves in the decays of these mesons. We don't produce them directly like CMS or Atlas, but they have a very subtle influence. And this with our specialized detector, this is what we try and look at.
So you're kind of getting the results of things and actually trying to work your way back and try and prove what was actually there before, sort of like there's like, say, a car crash, and you're working back to work out how the car actually crashed. But instead of cars, you just got the fundamental particles of the universe.
I mean, this is what we do in particle physics. This is how we reconstruct what was really there. I mean, all you can do is you smash your protons together, stuff flies out, and you have to record all that stuff, and then what you have to do is trace everything back to the point of origin and work out what actually happened. And it's quite difficult, but a lot of fun.
So what do you do? What's your day job like as a PhD student looking into this?
Oh, well, So I deal with sets of data. We have, so over the last 2 years, we've been constantly taking data. So we've had proton collisions. They've spewed particles throughout our detector, and then we've worked out where that's hit our detector and read that back out to a hard disk or a computer drive or something. And then what I do is I look at that information, reconstruct the particles, and then work my way back to what actually happens. So I turn up and I have my computer, and I have a set of data, and I look through that set of data looking for what actually happened. Sometimes this set of data may be simulated data. So what we do is we use computers to simulate what might actually happen, so we can then compare it to what actually really did happen, and gain an understanding of whether what we think happens is what really happening.
So you've got so you've just got a model that you're testing of the world and seeing if the world corresponds to what you think it's going to do.
Exactly. I mean, this is how you do science. It's just our model is particularly abstract, and we need to smash quite a lot of stuff together at quite high energies in order to understand it.
Okay, well, thank you very much, Alex. Very informative. And if any of our listeners, watchers, have got any questions, you can post them on the YouTube video there, or on the Google Plus page. Our next section is Sonja's, and she's going to be translating a scientific paper into plain English for us. Sonja, what's your paper about? Sonja, you're having mic issues again. Can you check your microphone, please, Sonja? Still having the mic trouble there. Sonja, do you mind if you come?
Microphone issue?
There we go. You're back now. Carry on.
Excellent. Sorry about that. Anyway, so my paper is one that got published this week, and we're going to start with a bit of imagination. So, imagine for a moment that a mouse, that is, and sniffing around for food, you smell the telltale scent of a cat. Your worst enemy, so you run away and hide, and you live to squeak another day. Or not. If you're a mouse that has an infection by a parasite called Toxoplasma gondii, your reaction will be quite different indeed. Instead, you'll find that scent of a cat appealing and wander towards it right up until you become some moggy's lunch. And while that's not the ending the mouse would have wanted, it's exactly what the parasite wants. Because the next stage of its life cycle, and that's the important stage where it actually reproduces, actually occurs in cats. So while the behavior change was easy enough to notice, exactly how the parasite makes it happen was a mystery. But a paper published this week, that's the one I showed you at the beginning, solved that mystery. So, how did they do it? Well, in order to survive in the body of a host, that parasite has to avoid detection and removal by the host's immune system. T. gondii does this by hiding inside the very cells that are supposed to detect it. These cells are called dendritic cells, and they're a part of the normal mammalian immune system. So this study showed that instead of the normal immune response, the parasite hijacks the dendritic cells and causes them to make and then secrete into the environment around them a chemical messenger molecule called GABA. And then this GABA activates the receptors that detect it inside the cells that have been infected by the parasite T. gondii. And that activation, they found, causes the cells to become more mobile than usual, which transports the parasite around the body faster and allows it to reach the brain and other parts of the central nervous system, which is where in animals infected by T. gondii you find large buildups of the parasite. But importantly, GABA is actually a neurotransmitter. That is, it's one of the molecules used to regulate our neural cells in our nervous systems. And its normal function in our brain is to inhibit receptors which allow us to reduce feelings of fear and anxiety. It's also the GABA receptor that alcohol acts on, having a similar effect to GABA itself, and thus likely causes drunk people to become more reckless than usual. So it's actually a relatively logical step to then see why a brain that has a high amount of these parasites, these T. gondii parasites, that are causing an increased production of GABA, might then cause the animal to become inappropriately fearless. And the author of this study says it's hard to know which of these two effects came first, its ability to evade and in fact use the immune system, or to cause behavioral changes, but the combination clearly gets the parasite to where it wants to go. So that's a little bit of up-to-date pathogen biology for you from this week. And back to you.
Thank you very much, Sonja. Scary parasites there. It's not nice to think of parasites being able to affect your behaviour, but obviously in those mice it does. Thank you very much for that. Now we've got, finally, Emma, who's going to be giving us a beginner's guide to corrosion. Emma, what do we not know about corrosion that we really should?
Well, most things. So the first question is, what is corrosion? And simply, it's a chemical reaction that causes metal objects to lose metal from the surface. Now, if you know anything about corrosion at all, you probably know about rust. And rust is just a special name that we give to the corrosion of iron. And rust is a big problem. About 1/7 of all the iron that's made every year is made just to replace the iron that's corroded away. So let's have a look at how rust is formed, and then I'll show you a clever way in which we can stop rusting happening by rusting something else. So I've got here an iron nail, nice and simple. And here I've got a tube of salt water. It might not look much like salt water because it's a bit yellow, but that's just because I've added an indicator solution, which is going to show us what's going to happen to the iron nail as it corrodes. Just pop that in there for a second. Leave it there. Now the reason why I'm using salt water is, well, I could have used normal water. That corrodes iron too. But salt water, the salt in salt water is much more aggressive, and so it corrodes it away much faster. And that's something to think about if you're driving around on the roads this winter after they've been gritted with salt. It stops the ice forming, but it means instead you get a salty slush, which is going to spray all over your car and corrode it away, so don't drive too much. Alright, let's have a look at how this has been going on. And you can see that there. Hopefully you can already see we've got quite a few bits of blue on the iron nail surface there. And what that means is my indicators change colour because we can detect iron is now dissolved in the salt water. We've corroded the nail. Bad stuff. And I've got another nail here that has been in solution for a little bit longer. So it's been in the salt water about 5 days. I'll just get rid of the bar actually so you can see it better. There we go. This has been in there for about 5 days. So just set it in a gel so you can see what's going on a bit more easily. And you can see you've got lots and lots of blue all around it. We've really corroded that nail away quite heavily. But there's something else going on as well. When we corrode things, we don't just lose the metal. We have another reaction occurring too. And that's because when we corrode something, we make electrons move, just like electrons in a circuit. So here, we create some extra electrons which travel down the nail to this pink region, where they react with oxygen and water in the salt water. And while that reaction is happening, that part of the nail cannot corrode. It's actually protecting the nail. So now, if we can control the movement of electrons, we can control what corrodes. So I've got here the battery connected to 2 nails. I'll just complete the circuit by adding some more salt water in and we'll see what happens. So hopefully, you can see, I'm not sure it's very clear, but this nail here, we're getting all the pink generated. And that pink is the protection reaction, so that nail is protected from corrosion. Whereas this nail here is blue. We're corroding that one away more quickly. And this is actually how they protect pipelines under the sea. They get a piece of scrap metal that they don't care about and they make it corrode really quickly by attaching it to a battery. But in doing that, the protection reaction is all that occurs on the pipeline itself, and so it doesn't corrode. And so in that way, we use corrosion to prevent corrosion. Back to you now, Michael.
Oh, thank you very much there, Emma. And we haven't had any questions in, but which means that's it for us. So I'd like to thank Alex for being our interviewee for this week.
Cheers.
And I'd like to thank Sonja for her scary parasitic paper.
Thank you.
And Emma for that beginner's guide to corrosion. We'll be back with another show sometime in the new year. So if you'd like to listen to that, you can follow us on Google Plus and just type in Sci Cam, or on Facebook. Again, just check Sci Cam. Thank you very much and have a good evening. Bye bye.